The Immortal Alchemist. It was year 2023 in the quiet hills of Switzerland. Dr. Adrian Sylas, a once renowned cellular biologist, found solace in his secluded laboratory. His obsession with the Turritopsis dohrnii, the immortal jellyfish, consumed him, driving him to the brink of madness in his pursuit of cell or organ rejuvenation and eternal youth. This creature possesses the remarkable ability to revert to a polyp state through a process called transdifferentiation. When faced with environmental stress, physical assault, or simply the natural aging process, the jellyfish can transform its differentiated cells into new types of cells, essentially starting its life cycle anew. Inspired by this, Dr. Sylas hypothesized that, if he could understand and replicate this process, he could induce a similar effect in human cells. His research led him to identify a set of proteins and genes that were key to the jellyfish's transdifferentiation. He then worked on creating a serum that could induce the expression of these genes in human cells. His fascination led to a groundbreaking discovery: a serum capable of reversing cellular aging. As the world outside buzzed with the monotony of daily life, Dr. Sylas delved deeper into the mysteries of the jellyfish, desperate to unlock the secrets hidden within its translucent frame. Nights blurred into days as he pored over ancient tomes, dissected specimens, and brewed concoctions in a desperate bid to defy the relentless march of time. But amidst the sterile walls of his laboratory, Dr. Sylas found himself grappling with the weight of his obsession. The faces of loved ones, lost to the ravages of age, haunted his dreams, their voices echoing in the hollow chambers of his mind. He made a serum called the chrono-elixir. It contained synthetic versions of the jellyfish's proteins, mimicking the Yamanaka factors. These proteins could activate specific genes in human cells, triggering a cascade of events that would rejuvenate the cells without compromising their specialized functions. Telomeres, the protective caps at the ends of chromosomes, naturally shorten as cells divide and age. Adrian Sylas's serum included telomerase activators enzymes that could restore telomere length. Longer telomeres meant healthier cells, an extended lifespan. Aging involves changes in the epigenetic markers that affect gene expression. The chrono-elixir aimed to reverse these changes, restoring a youthful gene expression profile. It was like hitting the reset button on cellular aging, and it can make failed organs to rejuvenate or reanimate. But when cells stop dividing and become harmful was a roadblock to rejuvenation. The chrono-elixir included compounds that prevented senescence, keeping cells youthful. With trembling hands, Dr. Sylas mixed the serum that would become his life's work, the chrono-elixir. Its iridescent glow held the promise of immortality, but beneath its shimmering surface lay the darkness of his own mortality. He ventured in the streets in the night searching for his guinea pig. He managed to lure a homeless and middle-aged woman named Emma to his laboratory. He made her sit in a chair, which was like a sofa. As soon as he gave her some milk to drink, she fell asleep. He then started his experiment on her. He injected his serum in her, but it was not a success because she experienced severe allergic reactions to the serum. Her body, unable to tolerate the foreign compounds, went into a state of shock. Despite immediate medical intervention, Emma succumbed to the adverse effects. Her tragic death made Dr. to think about ethical concerns about the risks associated with experimental treatments. Furthermore, he grabbed some more opportunity to lure some more volunteers into his lab, who developed abnormal cell growths and tumors after receiving the chrono-elixir. It appeared that the serum's potent effects on cellular rejuvenation also increased the risk of uncontrolled cell division, leading to cancerous growths. Dr. Sylas was devastated by the consequences of his creation. Dr. Sylas returned to the drawing board, meticulously analyzing the serum's components. He identified the elements that caused abnormal cell growth and began modifying the serum's formula to mitigate these risks. He implemented several safety measures to prevent the adverse effects observed in previous trials. Selective growth factors. He adjusted the concentration of growth factors in the serum to promote healthy cell division without triggering uncontrolled proliferation. Targeted delivery. Dr. Sylas developed a targeted delivery system to ensure that the serum's rejuvenating components were delivered directly to aging cells, minimizing the impact on healthy cells. Improved DNA repair. He enhanced the serum's ability to repair DNA damage, which is a common cause of cancerous mutations,